Ken Duffy is an old friend and colleague from Hamilton Institute in Ireland. He has mathematics degrees from Trinity College, a PhD and bachelor's. And he's a very modest CV, considering his amazing contributions to science. And I won't go through them all, but just, recently he became involved in biological applications. So he's been, we know Ken from applications to communications and to statistics, but in recent years has become involved in biological systems in medicine, and has at least two recent articles in Science. And just listen to these titles. Activation-induced B-cell fates are selected by intracellular stochastic competition. That's Science 2012. And an algebra for immune responses is Science 2014. And this is serious work, so he has new collaborators in Melbourne. It's very exciting. So, but here we'll hear about something different.
So thank you, Sean, for the introduction. Thank you, everybody, for coming to what is clearly a mystery talk. I was going to start with an unusual tone. It's going to be an unusual talk anyway, right? And the unusual tone is to point out that for me personally, our hosts, Sean and Muriel, are really trendsetters, right? And you might think that they're trendsetters for me because I happen to have collaborated with both of them, which is true, but it's not what I really have in mind. What I have in mind is that if you're lucky, once a year you get an invitation to Florida to come to this fantastic 3C meeting, but it comes at a cost. And the cost is an instruction on what you must do on research that year in order to qualify to come, right? And, I know a lot about clouds. I'm from Ireland. We have a lot of that. I don't know about electricity. We've yet to get electricity in Ireland. So I saw this year, and I thought long and hard about it, particularly bearing in mind that it's about 13 degrees warmer here in Florida than it is in Dublin at this time of the year, and you get about 4 hours extra sunshine. But I just couldn't bring myself to do it. Smart grid, green stuff, whatever. So I'm not going to talk about that. You can hear about that stuff from the other experts in the room. Instead, I'm going to tell you about something totally different. I'm going to go so off topic, I figured we're going to go all out. We're going to go completely off topic. And so I'm actually going to talk about biology, which I didn't talk about previously on being here. And the reason I want to talk to you about biology is effectively, you already know that statistics and engineering and computer science has made a huge impact in bioinformatics. And my claim to you is that the reason that occurred was the technological innovations facilitated it. It became very quantitative. You got a lot of data and a lot of stuff to dig through. And my claim to you is that same whole process is happening in cellular biology at the moment. And by pure good fortune, I stumbled upon it in 2007 when I met this man, Phil Hodgkin, who's one of my long-term collaborators now. And with Phil, I've had the good fortune of publishing, as Sean said, 2 papers in Science at least. But just to limit my ego somewhat, I'll point out that the first time we got beaten to the cover by kelp, and more recently by a brick. So, it's good to keep an element of modesty. And so, I'm not going to tell you about either of those 2 pieces of work today, and as Sean did indicate that from the titles, which seem probably a little obscure to this audience, the reason that we're kind of engaging in this work is that 10 or 15 years ago, if you were talking about cellular biology, it would be quite a qualitative description of the theories. What you would be saying is, if I add signal X, I get more of Y. If I add something else, I get more of something else. But what's happening is that there's huge technological innovations occurring on the experimental side that's leading to very fine, detailed information, and that facilitates a quantitative design and a quantitative analysis. So people in this room, there is an opportunity for you all to contribute, if you'd like. And so what I'm going to do today is, I thought I would give you, I'm not going to tell you about either of these pieces of work, but I thought I would give you a kind of a little hint of something that fascinates me. As Sean indicated, I'm a probabilist to begin with. I'm going to give you a hint of that. I'm going to give you a hint of the technologies that are coming along that are revealing this information that I'm claiming is more readily available. And then you can't come to a meeting that Sean organizes without theorems. Sorry, theory. There's a rule, right? Or certainly if you're one of his collaborators, you can't. So there will be no epsilons in my talk because I've substituted them with p's. And so, after doing some motivation and describing some things, in the latter part, I'm going to tell you about some work that we're presently doing that I'm enthusiastic about as a mathematician. And the reason for that is that it's the first instance where I'm working in this space by looking at the kind of questions my collaborators had. It led us to question something in an abstract mathematical sense that led us to prove some theorems that in turn is leading back into the biology. And so I thought I would tell you that story. But I want to start by telling you a little bit about cellular biology. And again, we only have so much time together, so I'm giving you quite a high-level view, lying a little here and there. But if anything I say is at least unclear on an elementary level, just interrupt me. I really would prefer to be didactic, okay? And so one thing that nearly all forms of cellular biology have in common is, and we can use the adaptive immune response as an exemplar, is that you start off with cells, individual cells, that have multiple potentials. And often they divide and segregate and specialize. And kind of the process of how that happens is fascinating. I don't know if you know much about about how your immune system works, but there's effectively two components to it. One component is very old, and plants and everything possesses it. It's called innate immunity. It's kind of the fundamental first line of defense. The more sophisticated part is called the adaptive immune response, and all jawed vertebrates share it. And the adaptive immune response is fascinating, and for this audience I thought I would at least tell you a touch of why it's fascinating. The way it works is within your body you have millions of these objects called lymphocytes, little individual cells. And if I was to take two from Vince, what you would see is on the cell surface, one of them would have this particular receptor, and the surface of a cell has loads of receptors by which it sees the outside world. And you might find 40 or 50,000 copies of one particular receptor, which is the receptor with which it sees the outside world. And if I take two cells from Vince, with very, very high likelihood, that receptor will be different on each of those cells. And the reason is, the way biology works is the interactions are complementarities of shape, not of chemical binding. So the way you see the outside world is I have a shape, and if I get good physical connection with the shape of my receptor and the shape of something else, then it triggers something within the cell. And so what happens in your immune system is you have millions and millions of these lymphocytes, and each of them on their cell surface has a receptor. Should something foreign come into your body and that get a good bind, it's a warning to your body that there's something foreign in there and it needs to react. And so the way the adaptive immune response works is I now have a small number of objects. So if you get say a bacterial infection, you might find that 20, 100-ish cells in your body can recognize this foreign componentry. And now you only have this small number of objects that can see this foreign thing, so what do? What they do is they get a signal to proliferate, to clonally expand, to make copies of themselves. And that fast expansion you've experienced firsthand. Whenever you get swelling in a lymph node, that's what's happening. You're getting this exponential growth in those cells specialized to see this foreign invader. But just seeing this foreign invader is not sufficient. At some stage they have to differentiate, change their type to become effector cells to fight the infection. And so if you take, for example, there's two arms of the adaptive immune response, T cells and B cells. We won't fret about it overly. But B cells, their effector state, how they fight the infection is they secrete small molecules into your blood. And they can secrete many different types. They don't secrete many different types. Each individual cell can only secrete one type. But somehow a decision-making process has to go on about which type am I going to secrete. Different ones are good for viruses, for bacteria, actually for worms. Usually right at the end of your immune response, you start producing a defense against larger pathogens. You normally start trying to fight viruses, which makes sense because they're the most damaging and risky thing on a short-term basis. Okay. But this kind of serves as an exemplar system. It's also a very, if you're interested in cellular biology, immunology is very nice because we know a lot of the triggers to kick off this process. And the way the process works is initially the cells do very little, and then you get exponential expansion. During that exponential expansion, some of the cells change type, and ultimately they clear the infection, and then the majority of them die off. a small number convert to what's called a memory state. They live in your body for a very long time, and next time around, should you get infected, they don't go through checks and balances. So these initial cells, the first time you expose them, they go through a lot of checks and balances. It's very dangerous to launch an immune response. You might launch an autoimmune one, but if I've already been through one and it worked out and I'm still alive, then next time around, why bother checking and balancing? And so that's what all of vaccination is based on, right? But it's a beautiful system to work with, in part because we know a lot of the triggers, and also because it takes place over like a 7 to 10 day period, so it's experimentally accessible. And if you take these cells and put them in a dish or do it in a mouse, what you find is you kind of find quite a smooth rise in the populations of various sorts, right? So as I said, you can take these cells can fight infections in different ways, and you see quite a lot of heterogeneity, quite smoothness, but it's repeatable across animals. And so one of the interesting questions that a lot of people are trying to address at present is, where is this heterogeneity coming from? It's not quite as simple as where's the decision making happening, because this evolution has brought us here. There's not necessarily decision making taking place, but how do you get that kind of level of heterogeneity? And there's a lot of potential sources. And one of the fun things about cellular biology is you'd be surprised at what we don't know.
I'm sorry.
Yep.
There's no exposure to environmental insult. It just does it.
If I do it in culture, depending on the type. So if you take B cells, you can stimulate them in one of 3 ways, all of which mimic things that they would naturally see in their body. One is exposure to CpG DNA. CpG DNA is basically a hallmark of bacteria. So if I take B cells and I give them a hallmark of bacteria, they'll expand and contract, but they don't take on an effector state. They don't actually turn into fighting cells. They expand and contract. You get the dynamics. The next thing along, which is also very old, LPS, which is a polysaccharide thing, you can expose them to that. They will expand and contract, and some of them will take on a fighting state. If you give them additional signals that they will get from other arms of the immune response, they start to differentiate and they become fighting cells. And then the last thing, which is you interact with them through something on their cell surface called CD40, cluster differentiation 40, that mimics interaction with the other major arm of the immune response, T cells. So if I expose these cells to these stimuli, I don't have to do all the complicated interactions. If I expose them to these stimuli, which I can do in culture, and I add in cytokines of various sorts, then I can encourage all of these final behaviors.
So the point is there's some intrinsic, I'm not going to say chaos, but you know what I'm kind of getting at.
Yeah, right. You know, I'm your kind of nut, so we'd be on the same team on this one. But it's not the only one. We'll get to what's going on. And what I meant to say earlier was, one of the intriguing things is, I said, you have millions of these cells and they all have a distinct receptor. How do they do that? How they do it is because your DNA codes for a stochastic program. It was hypothesized in 1959 that was the case by the great Macfarlane Burnet, who is where my collaborator Phil works. Phil now has his job. When Mac Burnet was there, he hypothesized that the only way you could produce that sort of coverage is by having a kind of decentralized stochastic program that made these receptors, availing of combinatorics. And that is actually what happens inside you. It was then shown provably in the 1980s by Tanagawa, who went and got a Nobel Prize for it as well. Okay, so there is a role for randomness, right? But the question is, where is this heterogeneity coming from? And there's potentially loads of different sources. I'm going to show you some examples of how we see this heterogeneity in a little bit. The first one is that the initial cells, they may have been different. The way you classify cells in biology is by looking at the cover of the book, basically. You look on the outside of the cell and you go, I'm going to classify things that have a lot of something and a little of something else as being in one group. Sometimes they're quite functional assays. If we take a collection of cells and you've got a lot of this receptor, and then I separate you and I expose you to something, you do something. So I'll say, okay, everything that has that receptor is all kind of correlates. I'm going to call those one cell type. So it could be the original cells were somewhat different to begin with. It could also be that when I initially expose them to some stimulus, how much stimulus that initial cell sees programs what happens later. When a cell goes to divide, the first thing it has to do is unzip its DNA and make a copy. And the new DNA in the cells that are born, what's open and available to be read and transcribed, may not be the same as the mother. And so that you could induce heterogeneity at that stage by changing what you expose of the program at the next generation. Partition is a particularly interesting one. I don't know if anyone in the room has ever wondered about the good fortune that you have feet at one end of your body and a head at the other. Now, some people tell me my brain might be somewhere else, but there's something very curious about that. You started out as an object that had no direction, right? But you have direction. And how does that occur? How that occurs is by a process called asymmetric cell division, the chemistry of which is not well understood. I'm going to kind of repeatedly say stuff is not well understood to tempt you in. Okay? The chemistry is not very well understood, but the principle is relatively simple, and some of the key players have been identified. So what happens is, early on, before a cell goes to divide, it produces these objects called polarity proteins, and it segregates the content of the cell. And so prior to division, the thing looks like a peanut kind of shape, and I'm going to get division down the middle. What happens is you segregate the content of the cell so that what's on one side and what's on the other is not quite symmetric. And now when the division occurs, the two objects are born with different content, and it becomes self-reinforcing. And so from those early cells, there's arguments about exactly when it happens. In the fly data last year would have suggested it's at about the time you've got to 16 cells. By the time you've got to 16 cells, from kind of there on in, you're starting to have this asymmetric cell division. It forces you to generate a spine, and then everything kicks off it.
And that's cell state.
Cell state. That's cell state, right? Again, like this, talking to this audience is kind of interesting as opposed to talking to a biology audience, but you are thinking about it in terms of how would you exercise control in these systems, right? So that's partition, and so there's a question actually, particularly in immunology, as to whether partition matters, whether the heterogeneity comes about from it. You can imagine if you have very small numbers, partition is essential, right? Which is early development, partition is essential. I don't have room to say 50% of the time I'll make this, and 50% of the time I've got 16 cells. I need to make, I need my feet down there, right? So the next one, process. The actual process leading you to change your cell type. So B cells, when they fight, for example, they take on a state where they secrete antibody. In fact, the antibody that they secrete is a free-floating form of the receptor that they had on their cell surface. That receptor, the B cell receptor, was capable of engaging with the foreign pathogen. So what they do is they become factories for secreting that stuff into your blood, and then it circulates and should connect on top of whatever that pathogen is. But there are different types of antibody that they could secrete, and the way that they do it is they chop out part of their DNA and discard it, right? So you can't go back, you kind of decide this is what I'm going to make, and you chop out the other things. The processes that lead to that are complicated, involve a huge number of reactions, and you could imagine maybe nature has evolved to take advantage of the inherent randomness that occurs in those processes, and so that you have some sort of stochastic element built in. The last thing, which is the most disastrously horrible, complicated one, which again a lot of people will believe is the dominant one, is external influence. These things secrete small molecules that communicate with each other. We might hear a little bit about that from Chris later, maybe. They secrete small molecules. They even do mad stuff like autocrine signaling. They secrete stuff, and then they sense it and go, oh my God, there's stuff in the atmosphere. I should secrete more of it. So one of the dominant control signals for lymphocytes to expand, T cells to expand, is IL-2, and they secrete and absorb their own stuff. It's very odd. So external influence clearly matters because you get different signals in different places, but even if you do reductionist things where you remove the external influences and you do it in a dish, you get a huge amount of heterogeneity. And I have individual friends that believe each one of these things is the dominant force of heterogeneity. And normally I would be put into the bucket D, which is not surprising given I'm a probabilist, or more recently A and D. But the reality is often you get a lot of redundancy in biology, and who knows. But the beauty is, while there's controversy, and there is, One, you can argue about it in public, as we have done with our pal Steve Reiner. Steve Reiner strongly believes partition is responsible for a lot of the diversity, whereas we're always defending randomness. But the real beauty is that this is to all of our benefit, and the fact that people have different hypotheses is to everybody's benefit, because people are designing different experimental systems expecting to reveal that their thing is dominant. And the reason why I'm in category A and D now is that I used to be in category D, and we set about designing experiments, and it revealed that A definitely had a role. A being that very early signaling was influencing the first cells, and that was inherited in their family tree, right? So that there was familial dependencies that we didn't predict.
I'm sorry, when
Yeah.
you said you're in A and D, you mean you believe those to be the dominant ones, but the others can still exist?
What I mean is that people would anticipate I'm a defender of A and D, and I have first-hand evidence that I know A and D are significant.
But you're not saying B and C don't?
No, absolutely not. Well, I'll do so in public in order to get additional funding, but everybody's agreed E is important. Yeah, no. And partition is one I'm suspicious of. The reason I'm suspicious of is the early studies. What they did was they tried to show that this asymmetry was happening in cells, and it's very hard. It's very hard thing to measure. And a friend of mine, who's now a lab head in WeHi, when he was a postdoc, he set about doing the very sensible thing of saying, okay, I don't really care if I see a little bit of asymmetric cell division. What I want to know is, does it have a long run effect? And at the time he was working in a lab, the firmly believed partition was important. And so what he did was he made a series of mice. You can't completely knock out these polarity proteins. So life is good. You can take part of the genome and discard it and make an animal without that part of the genome. If I completely remove a polarity protein, it doesn't grow, because you need it to generate the structure when it's born. There's a huge amount of your DNA is repeatedly used. What he did was he made knockdown mice. These are ones that produce very little of these proteins, enough to get through development, but not enough to see this kind of asymmetric cell division. And those mice are perfectly happy and healthy, and their immune responses look the exact same as a normal mouse's immune response. So that's a very fine functional demonstration that perhaps it's not so important. But it doesn't mean it doesn't take place. It just means maybe there's some redundancy in the system. I don't know. But the main thing is just the heterogeneity is itself fascinating. And I want to tell you a little bit about what do I mean by this heterogeneity, and what are the tools that are leading to observations about it, and why are they quantitative, and why is there space for us? Okay. And then I want to get on to some really ugly math, because I can't come to Sean's meeting and not show some ugly math. All right. And so the first thing I want to tell you about is I said, well, these things are heterogeneous, and you've had to believe me. So I told you, I take a load of cells and I put them in a dish from the immune system of a mouse, and I expose them to a danger signal, and they go about expanding. Okay. And I said it looks nice and smooth. There's a very beautiful technique which was introduced in the mid-1990s that is now basically standard, but my collaborator Phil was heavily responsible for showing demonstrated important hidden variables in the decision making of lymphocytes. And so the way this works, let me explain. I should have had I don't have a whiteboard to show you, but let me try and explain verbally. The idea is that I take my initial cells, okay, and I stain them with a dye. It's a green fluorescent dye. It has no ramification for the cells. Cells don't know about it. It doesn't make any difference. But I stain them all with that dye. And you actually stain them with a lot of it. And when they divide, their daughters inherit about half. Okay? So the initial cells, see this peak here? These are cells from the immune system of a mouse after 3 or 4 days post-infection. And what you're seeing here is this spike. Each point in here is a measurement of how many cells we saw that had this level of fluorescent intensity. These are cells that haven't yet divided. So that if they divide, their daughters have about half the fluorescent intensity. This is a half again, this is on a log scale, and a half and a half and a half. So cells in each one of these slivers have been through an additional division. Okay? So the first thing to note is this is after 3 or 4 days, you have a chunk of cells that haven't divided, and then you have cells in first, second, third, fourth, fifth, sixth divisions, right? So there's a huge amount of heterogeneity in the speed with which these things are expanding. Okay? People are still with me? All right. And so the vertical plot, so this is done by a technique called flow cytometry, which is in every lab, right? They're objects that are about the size of a small photocopier. They're not big at all. And what they can do is you can take cells and you stick a pipette, or you stick them beneath a pipette that sucks them up, and it can measure like 50,000 a second, and it can measure 10, 12, 13 different fluorescent colors at a time. And so in this case, for example, on the vertical axis, what you're seeing is each dot is a cell. We're getting a measure of its CFSE, this dye that tells you what generation they're in. So these vertical strata are again the generations. But on the vertical axis, what you're getting is a stain for something else. These cells, when they become fighting cells, they upregulate a sugary protein that appears on their cell surface, and I can look for it. And so what happens is that there's a huge amount of heterogeneity in the population, but they become fighting cells when they get to later divisions. So, so what happens is if I look at this at a population level, it all looks like it's coming up nice and smoothly, but in fact what the hidden variable here is that once you get through a number of rounds of division, then you start to become a fighting cell. Okay?
The 30-second physics of how you differentiate.
How you differentiate?
This,
Okay. This is.
Literally 30 seconds.
I'm going to tell you two different things, right? So here what happens is just when you change your cell state, you start running a different part of the program, right? And so syndicam-1 is just happens to be one of the proteins that you don't normally make, and you do when you become an antibody secreting cell. Okay? But there are other techniques. There are other techniques which I'll show you in a minute. And that kind of brings us on to the next one, and it will become a little bit clearer. Okay, so the reason that I said the stochastic element would be one of the things that I'm associated with is because that's what Phil strongly believed when I met him first and we were doing this work. And so, including, I thought I would mention him because he's not in the room, but Vijay Subramanian, who was a student of Bruce Hayek's, seeing as Bruce is, given the Illinois crowd didn't actually show up, I thought I could at least indicate there was some influence in here. Vijay and I got distracted into this in 2007, and then I just stuck with it when Vijay went off and did other things. But this is like getting population level information, quite a lot of population information. It's like hands up if you're happy at the start of my talk and at the end, and there might be 20% at the start and 20% at the end, but maybe it's a different group. You just don't know. And so my collaborator set about designing a time-lapse microscopy system to film these cells, right? So we could actually look at them. Now, time-lapse microscopy was used in the 50s with bacteria. I want to indicate to you how things have changed somewhat, okay? And so let me explain this very briefly. Hopefully the colors will show through. This is what you're going to see is you're going to see 7 days in the space of about a minute. You're looking at cells that are tiny. They're sitting in wells at the bottom of a big plate. The well walls are just designed to keep them physically a little bit constrained. They actually share medium above it, so there's this huge thick medium as far as they're concerned above it. It's, you're sitting in your little well, but you're not isolated from everything else. And these are cells taken from a mouse. Oh yeah, gigantic. Yeah, no, they're much less than a millimeter. Yeah. Oh yeah. These are very small cells. These are T cells, but these are taken from a mouse that was genetically engineered by people in the Riken Institute in Japan. Okay, and the genetic engineering of this mouse is such that, and this is one of the important techniques that allows us to look at them. Biologists have figured out how to put into DNA printf hello world, and by what I mean is these cells, when they're deciding not to divide, they make a red fluorescent protein. So they're sitting along, they're not dividing, they make a red fluorescent protein. What happens is there's a green fluorescent protein under the control of the promoter for division. So when a cell initiates the program to divide, the first thing it has to do is make a copy of its DNA. And so these cells, when they go about that division process, they make a green fluorescent protein. So while I'm in the process of going through the process of division, I'll be green, and when I'm just idly sitting there, I'll be red. So let me play you this. I should point out as well that this is a movie which Phil gave me from a paper that they had in PNAS last year that I wasn't involved in, but it's a great movie, and I thought you might like it. Okay, so they're initially red, they go yellow, they go green, they roll up, bang, they explode and divide. They're red, they go green, and off they go, right? They roll up, they divide.
Hello Kitty.
We're very unsure about the movement, actually. There's no real clarity about why they move. They do have a tendency to push into a corner before they divide. They roll up, and then eventually they go strong red, and then they die off.
But you were saying before the heterogeneity with division, and here it's not.
So Sean has made the point of this slide for me. Thank you. No, that's exactly it, right? So what happens is if I gave you a range of these videos, and I didn't tell you what came from what well, and you looked at it, you would see everything is nice and smooth, right? And so before we did the filming, our original supposition was there's lots of de novo randomness. And what happened was we went and started the filming, and we start finding there's huge within family concordances. So between different families, there's a lot of diversity, and if you didn't know any better, their overall diversity looks huge, but in fact there seems to be familial dependencies. So lo and behold, I've partially moved camp. Okay, so you're exactly right, and in fact here it's very, very obvious. The cells are all now dead, which is why they're just bouncing around.
What's the time scale here?
It's a week, yeah. 90, yeah, it's about a week, yeah. Normally you cut out the first 2 days because these are naive cells, so they've never seen an infection before, and so they ponder for a couple of days, and and then they get
all over the place, but then they go to the left when they just before they.
Yeah, we don't know much about the motion. Motion is not something that we've been. There's a few other things as well. This filming system is like 7 years in development, and I'm showing you nice movies. I could give an entire evening's lecture on outtakes, in part because there's something very beautiful with these cells, and I'm cheating a little, in that they stayed planar, and they didn't adhere to each other. And lots of them, the B cells, which we love so much, these are T cells, there's 2 different arms, they're both very important. What happens with B cells, which we've done some work on, and one of the science paper was based on films from B cells, is that they express adhesion molecules once they get going. And part of the reason that these wells were designed, they were built by a guy called Daniel Day in Swinburne University. He's a materials guy. Originally, even when we had these, you would have these beautiful movies where you'd watch the cells sitting in the well, they'd be expanding, and then this huge big tumbleweed of cells would roll in, pick them all up and roll off. And we've also been, we have a lot of ugly films. But the point anyway, the original point was twofold. One was to tell you that, so coming back to the question of how do you measure these things? So the promoter, the part of the program that gets kicked in when you become an effector cell, one of those transcription factors is known, it's called BLIMP1, and so there's a BLIMP1 reporter mouse. So BLIMP1 reporter means that I want to know when are you coding this protein, and what I do is in the DNA I insert a fluorescent protein afterwards, right? And the thing is, again, like making these genetically modified mice that have these kind of reporter systems, if it was 10 years ago, it would be 5 years worth of your life and an awful lot of shed tears, and you got to hope that you picked something right. Now you can make a mouse like this using a CRISPR system for like $40,000, $50,000 in maybe a 6-month period. So it's really, information is going to become available quickly. Okay, everything I've said so far, right, the things, or at least the 2 bits of data I've shown you, have been in vitro. And what happens if you work in biology, the first thing everybody goes, in vitro. It's nothing really like in vivo, right? In vivo everything is more complicated. How do you know anything you're learning? They are physically isolated.
That was disingenuous.
No,
there's nothing happen over the sides.
There's nothing happen over the sides, and so what happens with all of these things is we do controls that whenever you do this, you also take a culture where that's not the case, where we don't film them, because actually phototoxicity is also an issue, and so we get population level statistics, and you have to see that they're consistent. Yeah, so we do. The experimental guys are very good at taking care of those kind of things. so the next thing that people ask you about is, what about, where am I? In vivo, right? And so I'm going to give you one more piece of technology before we get to the ugly math. All is a taster, you know, not an extensive list. But so part of the problem is, if I want to know the contribution of distinct families, because we've decided families are important, but I want to do it within an animal, how do I do it, right? And so the normal way that you do those kind of experiments is by a method called adoptive transfer. I take cells from me and I inject them in Sean, right? But the thing is, I don't know, well, we're not related enough for that to work. Okay, but the thing is, you wouldn't be able to distinguish which progeny came from which cell. And so the technique, the idea is very old. Its current realization, you can probably attribute it to guys in Harvard in 95. The idea is that I'm going to make the cells a tiny bit genetically different, right? So I'm going to take these cells and not in a way that it impacts upon anything that they do, but only in a way that if I make their DNA a little bit different, then I can see that difference in later progeny. And you all know what a virus does, right? So a virus gets into your body, it finds a cell, it hijacks it, and what most viruses do is they hijack the machinery to make copies of themselves. But they're a very nasty form of viruses called retroviruses. And what HIV is an example. What a retrovirus does is it works its way into your cell, and then it copies itself into your genome, right? And so it copies its DNA into your DNA, and now every offspring of that cell actually has a copy of the code necessary to make that virus. As I said, HIV is an example. So what collaborators of mine, Ton Schumacher and people, have done is they've taken a retrovirus, they've stripped out its payload, so it still has all the machinery to work its way into the cell, to copy itself into your genome, but it doesn't copy itself into your genome. What they do is they put in a green fluorescent protein reporter so that they know that it's worked, and then they put in a little thing that they call a barcode. Originally it was like a 100 nucleotide snippet of DNA, kind of gibberish, but a sequence that you knew. And so they made a load of different little gibberish sequences, and so what I do is I take your cells, I infect them, this thing copies in this little barcode, okay, and then it goes off, I put it into Sean, it goes and develops, I pull a load of cells out of them and go, which one had which barcode? And then I can tell they came from a common progenitor. And the pace of development of this kind of thing is astounding. When Ton's lab had 2 papers, one in Science and one in Nature in 2013 using this, one looking at the immune system, one looking at hematopoiesis, which is the development of the blood system, which is crucial for post-cancer treatments. We can talk about that some other time over a beer maybe. And then there was actually a direct cancer study where people in a hospital took cancer off someone who had cancer, barcode labeled the cancer, and put that cancer onto humanized mice. So these are mice that don't reject human cells, and look to see whether there was a dominant progenitor developing that cancer. And Ton's library at the time, this is a couple of years ago, had like 2,500 different barcodes. And it was such a novel technique that they were getting papers in Science and Nature. And now his lab has a library that has millions of distinct sequences, and if you pick up the rate at which people are barcoding things now is phenomenal. Pretty much it just has become standard in the pace of about 2 years. Okay, and so the way it works is, of course, I barcode label your cells, I put them into a mouse or whatever, I let them develop, I sacrifice the mouse, which is the nasty euphemism in biology, and then what I do is I pull out cells of different type, and I can go, how many barcodes did I find, what are the barcodes, and look at the diversity. And the way nearly all experimental technologies develop is the first time people use them, they're quite descriptive in what they got. We use the technique, here's my data. It's like pretty ways of analyzing the data. And I was looking at this data from these sort of experiments with a collaborator of mine, Leila Perry. And one of the things that became apparent to us that launched us off on some mathematics was that we knew one of the things you don't get with this system is how you get the recovery is you have to sacrifice the animal. So I sacrifice the animal and I pull its cells out, right? And so what I can do is I see a snapshot in time of what's happened. But like the immune response I showed you earlier on, you initially have expansion and contraction and that kind of thing. And what we were thinking is, wouldn't it be nice if we knew how far down the family tree you were? But there's no obvious way to measure that, right? And so we went back, and now I apologize for the epsilontics, which will occur for the next little while. So I'd like to tell you what we did, which is effectively we went about a mathematical study for kicks. Let me see if how am I doing for time? Not too bad. How am I doing for time? 20 minutes, I'm in trouble. All right. Okay. All right. And so what we did, so I am going to skip a lot of the epsilontics then it would appear, was we went about wondering how would you measure the average generation of a family tree, okay? So cells initially start off in some generation 0, their daughters are in generation 1 and so forth, and what you have is you have a bunch of cells that are presently alive, and you'd like to know something about the average generation. It would give you a measure of the tree depth, right? And if you want to study things of this sort mathematically, you kind of have to go to branching processes. And branching processes start in a very important, totally politically incorrect paper in a journal that I presume you all read, which is the Journal of Anthropological Institute of Great Britain and Ireland in 1875. I don't have time to read this out, but I'm going to do it anyway. It's a great article, well worth reading, but you'll need a strong stomach, because it contains lines such that if that conclusion be true, our population is chiefly maintained through the proletariat, and thus an element of degradation is inseparably connected with those other elements which tend to ameliorate the race. It's
by the Reverend Watson.
The Reverend Watson and Francis Galton, yeah, so Galton being the famous eugenicist, or infamous eugenicist, depending on your point of view. But our story doesn't start, I would recommend reading it, it's quite funny. Our story really starts in, with a paper of Bellman and Harris in 1948. Kind of humorously, Bellman and Harris's results were announced in PNAS in 1948, and the paper appears in 1952 in the Annals of Maths, having been revised every year for the years in between. Bellman and Harris's model, and you'll see why it was important based on the year in a minute. Their model was effectively the following. It's a stochastic model of growth of a tree. And the idea is that an initial object has a random lifetime, and at the end of the random lifetime, they have a random number of offspring. The offspring themselves independently pick random lifetimes, and at the end of their lifetime they have a random number of offspring. And you know, it's clearly important in biology. If you do OR, you come across these things. They're kind of natural structures appear all over the place, but of course the reason that people were interested in them at the time was partly as a model of nuclear fission, right? So bombs. And part of the reasons their results were so important is the obvious question that you could ask was, what's the doubling rate, if you like? How fast do these things expand? If on average you have more than one offspring, you would imagine that on average I get some growth. And your back of envelope calculation would say, well, if Z of t is the number of objects alive at time t, then, you know, every average lifetime I get a multiplier of the average number of offspring, right? And that would be what our back of envelope calculation would do. And if that was your back of envelope calculation, you'd be wrong. And in fact, the doubling rate is necessarily larger. And they dubbed that rate the Malthus parameter. For the probabilists in the audience, if you haven't seen it, which you probably have, what you do is you look for the solution of where the moment generating function of the lifetime distribution with the negative argument becomes 1 over the average number of offspring. Okay? Don't have time to go into this at length because I've rambled on too much already, but I do want to at least say branching processes are very beautiful and interesting, at least from one point of view, is that their phenomenology is totally different to things like Sean's beloved random walk. In particular, if on average you have more than one offspring, whether your lifetime distribution is heavy-tailed or not, doesn't matter. And the reason why it doesn't matter, so you know, no matter what my lifetime distribution is, the reason it doesn't matter is if on average I have more than one offspring, the place where I'll have a solution here is for a positive alpha, so in other words for a negative element, right? So I've got a non-negative lifetime distribution, so I can definitely tilt the distribution left. Okay, and so what did they do? They said, well, if I solve this equation, kind of the trick in your mind of why would solving this equation tell you anything? The thing I want to hint to you is that this thing in here that I'm really taking an expectation of is itself a probability density. You're tilting the measure in a way that still gives you a probability density. And what it's doing is it's compensating for the fact that at the end of a lifetime you have an average number of offspring. And that's also key for why the long lifetime tail distribution doesn't matter. The thing that dominates your growth is the short lifetimes, not the long ones. When I have a short lifetime, I have 2 offspring, and then I've doubled my chances that I'll have a short lifetime. And later on, like Peter Jägers and Aretha and Kaplan proved that, in fact, this exponential growth rate, which they proved, right? So they said this alpha is my average population is growing like e to the alpha t, and alpha is my Malthus parameter. It's an almost sure result. As long as my population survives, it could die out early, but as long as it survives, ultimately every path grows exponentially. And the approximation is quite curious. It's not like an ergodic thing. What happens is effectively there's a random period of time before I kick off into the exponential phase. And you can think of it very easily, because I said this will work even if you have heavy tail distributions. So you can imagine that very first object might take a long time until it divides, and somehow I'll remember that. But eventually I'll have enough objects that I'll start getting short lifetimes, it will become self-reinforcing, and I get exponential growth. Okay, so I'm telling you that. I'm going to tell you one other thing before we move on, which is, and apologies because this looks brutally mathematical, is that normally people, we'll get to this, normally people don't write the Malthus parameter as a function of the average number of offspring, because normally the average number of offspring is just something that's in your model. And what I'm going to tell you is that the Malthus parameter is real analytic, which means you can Taylor expand it, right? And the way we proved it, I'm sure other people must have known, is just you basically just apply the real analytic version of the implicit function theorem, which thankfully is in one of Stephen Kranz's books. And it allows you to identify the derivative of the Malthus parameter, right? So as I change the offspring number, the Malthus parameter changes, and I can actually figure out how that change is occurring. I will say it should be pretty obvious that that's the case, because what I'm really doing is I'm looking for the intercept of this blue line and the red line, and as I move the red line up and down, where that solution occurs is nice and smooth, and it's not too hard to convince yourself that that would be true. So why do I care about this? Well, I care about it because I'm going to make one more weird observation before I get back to average generation. I care about it because I might get at the derivative of the Malthus parameter by the following method. If I started off with some cells that were labelly positive, and this label served no purpose other than just to identify cells, and with small probability I lose the label at some stage in my lifetime, and then once it's lost, it's gone for good. Okay? Well then, the average number of offspring of the big tree would be something, my H from earlier on, and the average number of offspring of the label positive tree, that is the subtree with the pluses, will be the average number of offspring times the probability that I don't lose the label, effectively, right? And so why would I care about that? Well, then I can apply these old results, and they tell me if I look at a large time and I have a small probability of label loss, then the growth rates of the two populations, the difference between them looks like some derivative of the Malthus parameter. Why do you care? Why would you ever care? What is this man telling us about? The reason is that this thing, when we analyze average generation, this darn quantity will be the exact quantity that shows up. And so if I could measure this quantity, I could draw inferences about average generation. Okay, so let me tell you that very quickly. So back to average generation. So now I'm labeling my trees by their generation, and I want to know something about the average generation of all living cells. And so the only way to do this is if G is the total generation, so 3 plus 3 plus 2 plus 2, I have to kind of jointly study the total generation of living cells and the total population, because they're dependent on each other. If I want to know something about how the total generation evolves, I need to know something about the total population size, right? And I'm not going to explain this in any detail. The reason why I wanted to put it up was merely say, if you use a trick that goes back to the evil Reverend Watson, so it's a very pretty paper. He had the key insight. The key insight is, I want to know something about how, the probability of the total number of objects evolves. And the system is kind of disastrously complex if I try and look at it that way. And he said, look at a transform. So take the Laplace transform of your distributions, which for us is moment generating functions or probability generating functions, then you can solve the problem in that way. And the trick is twofold. The first thing you do is I've still got a problem. I've got an average over all possible sample paths. The trick is I explicitly average over the experience of the initial object. And then it gives me a recursion. All right? So that's the trick. The trick is I've got these complicated trees, but I say, well, let's say the first object lived to time t, and I know what the probability that is. Then at that time, I have whatever, let's say 2 offspring. They give me 2 independent branches of the tree. They look like the original tree, but they've got a shorter period of time to live, so I get a recursion. I get these, normally you get seriously ugly nonlinear Volterra equations, and it gets even uglier when you start worrying about average generation. And the reason you do is there's a beautiful cross term, which I won't dwell on, but the beautiful cross term comes from the fact that we knew total generation and population numbers had to be related to each other, and that cross term captures that. And I'll probably skip the mathy proofs, but what happens when you get something like this? So this says, I know how the moment generating function evolves. Then from that I can tell you how all the moments evolve by merely taking, I differentiate this equation, I get another equation, and then I go and solve it. And you can do some asymptotic analysis, and it turns out that while the total population, if you have on average more than 1 offspring, grows exponentially, the total generation grows like time times an exponential. And when you work it all out, we get exactly this quantity we had before. So I'm going to skip telling you how you kind of prove these sort of things. Yeah, although I will say that the second element, when you take these ugly integral equations, the trick to analyze their asymptotic behavior is to use theorems from renewal theory. And for this average generation, it turned out that we needed a really very obscure one that, as far as I know, only exists in 2 places. One is in the back of Søren Asmussen's book on Room probabilities, which goes to show the plain fluke of knowing these things, where it comments, here we're going to prove this theorem, but it holds a much greater generality. And he doesn't tell you where. And I emailed him, and he wrote back to me, because Søren's a great guy, and he said, I just presumed it held a greater generality. And then he pointed me to a paper that he had written, which was exactly what I needed, which is in Siam Review, which is helpfully called on Wiener-Hopf factorization or something. I would never have ever found it. Because normally the way renewal theorems work is you say, I have a function phi, I have a distribution f, and I have some function capital psi that's written in this form, and I want to know something about the asymptotic behavior. And normally you want to know about psi of t when t becomes large. In our case, it turned out we're interested in psi of t over t, and it turns out that's a much easier theorem to prove. Anyway, so I'm going to skip it and merely point out, okay, so you do the math, you do the analysis, and it turns out that so long as your label positive population continues to survive, if I have a small probability of delabeling, then the average growth rate of the generation is actually this logarithm of the ratio. So it's basically, the log of the ratio is really telling you the difference in the slopes. You still with me, Sean?
Yeah.
So at this stage, we're just having kind of fun, right? Apart from, now we start getting excited. The reason we get excited is this object on the right. I want to know the thing on the left. This object on the right has loads of experimentally desirable properties. The first thing is that it only depends on a ratio, the proportion of label positive to non overall cells. And that's something I can potentially measure. I can't measure total cell numbers. That's never going to work. Normally, when people study these systems, they don't allow death. But we could have had death in the system, and it doesn't matter. And the last thing is, of course, it didn't depend in any way, shape, or form on actually the life cycle distribution, which is not something we could measure anyway. And so what happens with it, just to give you a hint, is that if I start with a single object, and I use, and so here these are stochastic simulations where the orange are actually the growth rate of, or actually the average generation of these trees that you're producing, and the blue lines are what our estimate would be, right? And they're disastrous at the start, and then they stay kind of disastrous, but the slopes are right. And the reason, of course, is that thing I said to you earlier on, you have some kind of crazy behavior until you hit into this regular expansion, and it's really the regular expansion that the theorem is telling you something about. But as we all know, everything in life is normally distributed. You always lie to everybody and you say it's because of the central limit theorem. It's not. It's because of the central limit theorem plus something called the delta method, which says if you have something that looks asymptotically normal, any bloody function of that thing also looks asymptotically normal. And that's the case here. So what happens is if I don't start with one cell, but I start with several, here these are the exact same scale, but starting with 100 cells rather than just one, then what happens is, like, I'm kind of cheating. I'm already getting into that phase where I've got a nice amount of averaging going on. Then I get very good estimates, right? So we're all very excited. It's all working very nicely in principle.
What's P though?
P is my probability of label loss.
Can you estimate that?
I'm going to tell you in a minute.
Oh, okay.
Right, so we proved these theorems, right? And so I swear, originally we were just kind of curious about it, but it proved promising enough that I happened to have the good fortune to be a co-PI on a Blue Skies research project funded by the Human Frontiers Science Program. The people I did all this math stuff with is my postdoc Tom Weber, who's from Luxembourg. So yeah, there you go. And Layla, who's over here beside me. And I'm co-PI with Andy, who's a computer scientist from Drexel who helps us do image processing on movies, with Phil, who you met earlier on, and the remarkable Ton Schumacher. Ton Schumacher runs a lab in the National Cancer Institute in the Netherlands, and he does lots of this, his work I mentioned earlier on, lots of this groundbreaking science, but also actually his main focus is actually on clinical applications. And so you'll have a scary paper, I think, out early this year where there were 4 terminally ill people with cancer whose lives he saved. But we went to this meeting, our annual meeting, and we decided to pitch this idea that we could measure average generation. We suspected that Tom would be interested because there's lots of sort of deep questions you could answer with it. And we proposed a genetic construct that would allow us to do this stochastic delabeling, right? Because I need something that will stochastically delabel. And the idea was, you know that Fuki movie I showed you earlier on where the thing makes a green fluorescent protein when it starts to divide. The idea was, don't make a green fluorescent protein. What I'll do is I'll have all cells make a fluorescent protein, but every time they go through cell cycle, they'll make something called Cre. Cre was found in bacteria. It's how bacteria share DNA with each other, right? Because they need to evolve too. And what Cre does is, one of the things you can do is that Cre, when it's expressed elsewhere in your genome, you can have these things called LOXP sites. And what it does is it folds the DNA at the LOXP sites and chops it out. Okay? And so the idea was that we could put a fluorescent protein between 2 of these sites, and every time you went through cell cycle, there's a small probability that the Cre would cause the accession of the fluorescent protein. You would no longer have the DNA facilitating you making a color, and so you would delabel, and all your offspring would delabel. Okay? So we went and pitched this to Tom. We actually pitched 2 different things. We pitched something else that I'm not going to tell you about today, which is also good fun based on combinatorics that's now being built in Weihai. But Tom got excited. He's one of those guys, he runs a big lab. If he decides he's going to do it, he does it. And so he's sending off email during the thing. Layla knows him well, and so she'd actually ordered all the bits that we needed to make this construct beforehand. And so, you know, there's a keen sense of elation. We've proved these ugly mathematical theorems and we're making a difference. And that sense of elation lasted, I'd say, about 3 hours until I was on the train on the way back to Schiphol, when I suddenly had this dawning sweat of, I've convinced this famous guy to take a postdoc and a student and spend 6 months or a year building a construct on the basis that I've discovered that the Malthus parameter is real analytic. and this derivative appears in two different places. And I, of all people, know that lifetimes are not independently identically distributed. I showed you movies earlier on that we've analysed where these things are correlated. So there's a keen kind of sense of panic. And so perhaps do I have time to show you a tiny bit of math? Oh, I don't. All right. Yeah, okay. I'm going to go. That's okay? All right. I'll probably, yeah, okay. All right. So I'm on the train on the way back with Tom and Layla, and I thought about, because like one of the things that had been bugging me since we found the result originally was, there was no lifetime distribution in there. Like somehow I've discarded the lifetime because the delabeling is kind of a division-dependent thing. So maybe there's another way of seeing this. And so this is a much simpler derivation of a much weaker form of the result, which gave me sufficient confidence that I didn't warn Tom that I'd broken into a cold sweat. And so the idea is this. If I give you a single cell in generation G, there'll be no trees in this. We're going to disregard the trees. Then if it loses its label with probability p at each generation, what's the probability that it is still label positive given that it's a generation G cell? It's 1 minus p to the G, right? And thankfully, everybody in this room has used the fact that expectation is linear to save their bacon at some stage. And so that's what we do here. You take a collection of cells, and I'm not going to tell you their familial relation. I've got, say, Z cells, and each of them is in some generation. Then the average, and now this average is a bit tricky because it's an average over these different realizations of delabeling. This is why it's a weak result. The average number of cells that are labeled positive is just this, it's, we just figured it out. It's 1 over Z, this thing. Now, those of you who've been to C3 before will know that I think that isomorphisms are one of the most powerful tools in mathematics, and my favorite one is that E and log are inverse to each other, which I've used to great effect for at least one paper in transactions and information theory, and I use it again here. So why do I do that? Well, I take E and log and I do this, and I go, if I call log 1 minus p theta, then this looks like the moment generating function of some random variable. And what is that random variable? Actually, the random variable is something where you uniformly select one of the cells. That's all it is, right? And why do I do that? Well, we all know moment generating functions are real analytic and are really pretty. And moreover, if I took the cumulant generating function and I take its expansion around the origin, I know that I'll get 0 plus theta times the first derivative, right? Now the first derivative is actually the average generation of these cells. And so if I divide by theta and take it to 0, it's the same as taking epsilon, p, and it's the same as taking p to 0. And I know p upon log 1 minus p goes to 1. And so basically we've re-derived that previous result, except in a weird thing where I've averaged over all the possible realizations of this delabeling, right? But the point is that it's not something that's solely dependent upon having a branching process and its asymptotic properties and so on and so forth. Okay, and so what I'm going to tell you is that this is still not proper experimental data, but a realization to show you that this thing does not just depend upon the branching process assumptions. This is the family tree, the early development of C. elegans, right, which is a nice little organism whose early birth is totally determined by asymmetric cell division. And this is very repeatable. You can take multiple versions of this worm and film them, and you'll find that a lot of the stuff is very repeatable. I see it once in one, and I can make predictions as when I'll see a division in the other. The early part of life looks like something unzipping itself, if you know what I mean. It looks very deterministic. And what we did was we took this family tree. This family tree looks nothing like a branching process. There are correlations in lifetimes. Initially it expands very rapidly and then it slows down. It's really brutally different. But if you apply the methodology, and we imagined we had our construct and we could stochastically delabel, then it turns out that we draw accurate inferences, right? So we're quite hopeful. And so, of course, the reason why we want to do this is to do with if we can measure an average generation in vivo. I'm also kind of indicating we're interested in doing it by constructs. But actually, there are certain things in your genome, particularly microsatellites, that mutate with small probability every time your cells divide. And so there are kind of inherent things that we might be able to use without genetically modifying objects to determine these things. But like, for example, there was a paper in Nature only late last year showing that totally understandably, the average number of generations that a cell population goes through affects the likelihood that you get cancer. Various cancers appear after a lot of generations because they've had more opportunity for mutation. There's also things like HIV. What happens with that is it's a deep chronic infection where your body gets exposed to this pathogen. It causes your immune system to launch a very minor response, but it never gives it enough energy, never gives it enough of a signal for it to really kick off. And so it divides, and then one of the cells dies, and then it divides, and eventually after a whole load of divisions, your immune system's worn out, the viral load goes through the roof, and your immune system can no longer, the remaining cells are, they call it exhausted. They're no longer capable of responding. But the estimates of how many generations that takes are quite broad and unclear, and so there's all sorts of things that we could potentially do. And I don't know if this will turn out to be the universally useful answer, but we're going to go look at it. And of course the obvious question is, well, does it actually work experimentally? And basically, the only hostage I have over Sean and Muriel is that I'll tell you if you invite me back next year. I do have preliminary data. They made a construct, not the one I've shown you, but they have made a construct. It is in cell lines. If we can get it working in cell lines, which I'll probably know in a month or so, then they're going to go put it in a mouse, or make a mouse that has the construct. And with that, I'll just close by saying it really is a very entertaining space to be in. So if you're looking for something different to do, I'd recommend it. But I'm going to stop by thanking Tom and Layla, who I did all the math work with, and also Layla, who's helping to drive oversee the work constructing the construct within NKI, and then Tom Schumacher's group for having the ridiculous faith of trusting a mathematician, and finally Phil, without whom I would never have been working on biology at all. Okay, and I'll leave it at that. Stunned into silence.
With the C. elegans,
Yeah.
these weren't extracted out. I mean, they were developing into an organism, right?
Yeah.
So they're talking to each other.
This is time. Yeah, right. So well, it's not clear whether they're talking or not at that stage.
They're contiguous. They're
Yeah.
producing stuff.
I don't.
Do we know they're not talking?
No, I'm not going to say that. I'm just saying that it's not completely obvious to me that they are, but they might be. Yeah, I don't know what they're secreting. The early development stuff is quite weird because it takes a few hours to replicate DNA. So one of the things I never understand about this data is that the early divisions are all actually like 15-20 minutes. Don't know what's going on. Yeah, the way this data is extracted is actually they do 3D microscopy, and so eventually the population gets large enough. I think it's at about 6,000 cells-ish. At that stage, you can't make inferences about the lineage tree perfectly. Yeah.
And a mature organism has how many cells?
I don't know. I could tell you how many lymphocytes.
I thought it was mapped all the way through to mature organism.
No, it's not. No, I mean you're not. Well, certainly the lineage tree thing you're not able to do. No. As I said, we have trouble even with certain sorts of filming.
So you answered my question.
Okay.
I never got over my fear of your P.
You never get over. Oh, okay, grand. So let me tell you how we measure P.
It sounds hard.
It sounds hard, but maybe it will prove to be. It's not trivial, but it's not too. The reason it's not too hard is demonstrated by this, actually. So what happens is my cells are initially labeled positive, and so the method we're going to use is that division diluting dye I described to you earlier on. What we're going to do is you're going to take a huge number of cells that are labeled positive, and we're going to stain them with this dye. And we're going to know that they're in generation 0 because we've stained them. going to stimulate them, let them go through a division. Right? And after one division, a certain proportion of them will lose their label. Right? And the thing is, because I can tell from one generation to the next, I know you've been through one, there's an empirical probability that, and I don't plan to do empirical probabilities, we're going to use the Bayesian method that we've developed to draw inference. There is a bit of it depends on how small p is going to be. Okay, the theorem says p should be small, really, really small. If you go too small, what happens is the variance looks like 1 over p, and so I don't want it to be too small. And moreover, there's other constraints about, effectively what happens is the math is telling you want p to be as small as possible, and what that's saying is ultimately you'll get great resolution, but I want resolution over a shorter timescale. And so the p's that we're looking at the moment, and we do have an experiment of this sort, I have seen the data. I should also point out that we can start with like 1 million cells, and so we're looking at a p that's 10 to the minus 4, 10 to the minus 5. Okay, but I can take 1 million objects, force them through one division, and I want to get a good estimate of 10 to the minus 4-ish. Did you get it?
Yeah.
Hopefully that will work, yeah. But, yeah.
That would be fine. Okay. Any other questions? Fascinating. Thank you.